I guess we can get started. I didn't realize that the seminar had changed to 3 o'clock instead of 3:15, but it used to be when I was here before. Most of you don't know me. I'm Jerry Nemi. I'm a retired professor in the department. Now I can do what I want, where I want, but I don't get paid for it. That's all. I'm here because to introduce my friend and colleague, Matt Etterson. And I just found out about a second ago that where he grew up. I didn't know that. I didn't have that in his resume. But he grew up in Connecticut, on the East Coast. And let's see, education-wise, Matt has a very unique background. He has a math degree from Yale, which is unusual, but I've met many ecologists over years that have had math degrees, and many of them are just outstanding ecologists like Matt. What are you laughing about? After that, Matt had a number of positions, including one interesting one in Africa at the Democratic Republic of Congo. We were there for a couple of years. And then he went on to do his PhD work at the University of Minnesota in the Conservation Biology graduate program. And shortly after that, he came to Duluth, along with somebody that many of you already know, Julie. Let's see, Matt wears many hats now that he's here. He actually gets paid by USCPA, where he's a research ecologist. But he also has been very active in the IBS graduate program, where he has a graduate appointment. And he has been teaching a variety of courses in the department with an adjunct appointment in the Department of Biology. But as if that's not enough, he also has a lot of activity with Hotbridge, which he'll talk about today, where he serves on the board of directors at Hotbridge. And he also chairs a research committee at Hotbridge. And having been on the board in the past and being on research committee, I know that ends up being quite a bit of work, because it's an active board, not the kind you just go there and talk about things and then you walk away. But it's also a very good place to have interactions with, especially for biologists and interested in birds, as Matt is. He's got an outstanding record of publications. I counted over 50 peer-reviewed publications that he has, and that's outstanding in itself. So with that, I'd like to introduce my friend, Matt Etterson.
Thanks, Terry, for that great introduction. You can see what happens. You retire and everyone starts knocking off 15 minutes early on a Friday, right? Well, I am really grateful for the opportunity to be here today. I'm glad that Jerry mentioned all 3 of these institutions, all of which I'm very fond of here in Duluth. We have our local EPA lab, we have UMD here, and today is the Hawk Weekend annual festival for Hawk Ridge. So I'm going to try to tell you a very large story today, a story that's global in its implications, spans many decades. And so it's a much bigger story than Hawk Ridge, but you'll see on many, many slides the Hawk Ridge logo. And whenever you see the Hawk Ridge logo, no, don't flap or make bird calls, but that's an indicator that Hawk Ridge data have contributed to an understanding of the problem that we're addressing. Maybe we'll just look at this slide. Help. Oh, I got it now. I can get it. Oh, I can? Does it work? Yeah. We tried it earlier.
Never mind, it must have been you. Oh no, it must have been you. Never
Thanks,
mind.
I'll go with this.
Okay.
It'll work. Okay, well, my talk is really going to be in 3 parts. Wait till you see my data slides in this font, right? At first, I'm going to introduce raptors, birds of prey, talk about the setting in which raptors come into contact with environmental contaminants. And then I'll run through 5 case studies. That's part 2, the tragedy in 5 acts here, illustrating what the exposures were and what happened to the raptors that were exposed. And then finally, the real goal of my talk here is to get to part 3, in which we try to draw some generalizations and come to a more general understanding of these birds and their vulnerability to environmental contaminants. So let's talk about our characters. When we think about birds of prey, so birds of prey are in 3 orders. The Excitriformes, Strigiformes, and Falconiformes. This is a paraphyletic group. There's paraphyly all over the place here, and I'll walk you through that in a moment. I'm going to be talking mostly about the Excitriformes. These are kind of what we think of as the true hawks, the diurnal raptors. And I've got a few genera here, and I'm only listing genera, as you notice, because I think from an environmental contaminants perspective, it's useful to think about how these genera of birds are similar and different. Osprey, of course, is a monotypic genus. Then we have the soaring hawks, Pedeo, that's our red-tailed hawk, Swainson's hawk, broadwing hawk. You may see broadwings today, or you may have seen them today if you're up on the ridge. We have the sea eagles in the genus Haliaeetus. Our bald eagle is in that genus, but there are other sea eagles in the same genus that are typical study species for environmental contaminants elsewhere. We have the accipiters, our forest hawks. This is sharp-shinned hawk, Cooper's hawk, northern goshawk. And then we have our vultures, which should be split into 2 groups, the old world vultures and the new world vultures. And I'll come back to that point in a minute. We, of course, have the owls. I won't be talking too much about owls today. I don't stay up that late anymore, but they'll make a few appearances. And then we have the falcons, formerly considered monophyletic with this group, but it turns out they're a much more derived group, having diverged from the clade containing parrots and songbirds of all things much more recently. And you'll see that this matters from an environmental contaminants perspective. So let's look at this phylogeny. The Excitriformes are here shaded in blue. The main clade is this one containing most of the genera that I've just been talking about. And you see the New World vultures set apart, having diverged from the rest of the Excitriformes, maybe 58 million years ago. The Old World vultures, in contrast, are nested within this Buteo accipiter clade somewhere. The owls, looking like maybe their sister to this, are not. This is a paraphyletic group. This clade contains a whole bunch of other stuff that are not considered birds of prey. And then the falcons are somewhere way down here, much further down. So if we look at the current distribution, global distribution of raptors, we see that they're mostly a tropical group, not terribly surprising, I guess, but that is the center of diversity. And so the temperate zone has fewer species. The Excitriformes and Strigiformes by far contain the bulk of diversity, with Falconiformes being way behind. Tropical bias, and 8 species are known to have gone extinct since 1500. These are all island endemics, so pretty similar story to island endemic extinctions. So why do we talk about raptors as being vulnerable to environmental contaminants? So we have to take a brief interlude into ecotox here to define some terms. And it's because of these food web processes involving environmental contaminants that put raptors at risk. So we can define bioaccumulation as occurring when the intake of a contaminant exceeds elimination. And this is by all routes. It could be dietary intake, it could be dermal intake, it could be inhalation, any route by which you're taking in some of the contaminant is intake. The elimination is also all routes. Urine, feces, biotransformation, metabolism. If intake exceeds all those processes, then the contaminant's going to build up within the organism. Bioconcentration, similarly, is said to occur when the organism has a greater concentration in its tissues than in the environment or substrate within which it lives. And these two processes together conspire to produce biomagnification. So when you have a trophic system like this with many links, the contaminant gets magnified through these processes with each link so that the animals feeding at the very top of the food web receive the greatest exposure. So when talking about environmental contaminants, I think we have to start with the organochlorines and DDT in birds. These first were introduced to the US in 1994, subsequently banned in 1972 after causing an enormous amount of damage, certainly to birds. They are a neurotoxin. They're very persistent. If you look at these soil half-lives, that's 2 to 15 years. They are bioaccumulative, as we just saw in the previous slide, but they're not very acutely toxic. So this is the median lethal dose to birds. Does everyone know what an LD50 is? Hands? No hands? LD50. So the LD50 is the dose expressed in milligrams of active ingredient per kilogram of body weight of the animal. So the LD50 is the dose that'll kill 50% of the animals that are exposed at that concentration. So that's a lot that you have to give a bird to kill it. But unfortunately, DDT at least did a lot of other stuff. So subsequent to its introductions, people started seeing pretty sharp declines in raptor populations, especially on the east coast of the United States. You see, this is a map of the peregrine falcon population around 1970, extirpated from the Appalachians, virtually extirpated from the Mississippi River Valley, and severely declining in the west, but holding relatively stable in the Arctic. And the apparent cause was nest failure, reproductive failure, due to breaking of eggshells. Eggshells were breaking, they were getting much thinner. And with investigation, it turned out that DDT, and especially its metabolite DDE, was resulting in eggshell thinning. And so these graphs give you some, so it was banned in 72 by EPA, and then many populations have subsequently recovered, as shown by these graphs of what's called the Raptor Population Index here. I'm showing from two sites. These are peregrine falcon from Hawk Mountain in Pennsylvania and from Hawk Ridge here in Duluth. We contribute to the RPI. And don't worry too much about what the vertical axis means. Just interpret it as being higher means more birds, lower means less birds. And what you can see here happening from the 40s up through the 70s during the DDT era, peregrine falcons were declining pretty sharply. Upon banning DDT and many other organochlorines, peregrine falcon populations began to recover. And our trajectory in Hawk Ridge started somewhat later, so we really only see the recovery portion of that story. Okay, organophosphates. Also a pesticide. Organophosphates were in use at the same time that DDT and DDE were around. In fact, Rachel Carson in her seminal book also wrote about organophosphates. But they gained a lot more use, became more prevalent once the organochlorine pesticides were banned. And they do have some attractive properties. They don't bioaccumulate. They don't impair reproduction. But if you look at their potency, they are much more acutely toxic. They're not very persistent in the environment, again, which is really good, but they're much more acutely toxic, right? It takes much less of this stuff to kill a bird. This is a little bit over 500 times as toxic as DDT on an acute basis. And so as these things gained more and more use, we started getting more and more reports of birds allegedly killed by exposure to organophosphates. And 3 species in particular pinged in this particular data set. And you can start to wonder, well, 243 birds, that's an individual tragedy for 243 birds, but from a population perspective, does that really matter? Do you really need to be all that concerned? But you need to think that if these are the number of carcasses of birds that were actually found and reported, that's going to be the tip of the iceberg. And so there's going to be many, many more birds that are either not found or not reported. So that's the first part of that story. The second part, I want to focus on Swainson's hawk. So Swainson's hawk is gregarious, at least in the non-breeding season, tends to forage in flocks, often feeds on the ground. This is in the genus Buteo, by the way. And in the non-breeding season, forages primarily on insects. In fact, grasshoppers make up a main staple of its diet. And you can see, based on where it's going, so it's breeding in Western US, heading down through the Central American corridor, and wintering down here in the Pampas region of South America. And that is a heavily agricultural region. Here's a photo here with the map again, where these birds are spending a lot of time in and adjacent to alfalfa, wheat, corn, and so on crops where a lot of organophosphate pesticides were used. So this is just a few visits done in 1996 to some of these fields, and the authors found over 5,000 Swainson's hawk carcasses killed as a result of exposure to these organophosphate pesticides. The main culprit here is monocrotophos, that's MCP, but chlorpyrifos and dimethoate are also organophosphates. And so the countries down here, Argentina, Brazil, banned the use of monocrotophos. And the researchers went back the next year and found many, many fewer carcasses, in fact, only a couple. So that appeared to work. And so we can think about, well, was there a recovery in this case too? But I think we also have to ask, was there an effect? So here's the event, right, coming sometime in between these 2 years. And this is when the mortality event would have occurred. This is RPI data from this site here, where presumably a lot of these birds have to pass by on their way to the Pampas. And you do see a decline in Swainson's hawks from one year to the next, but that decline is no bigger than many other jumps that we see in that data set subsequent to the banning of monocrotophos in that region. So was there an effect? Maybe, probably. A similar site very nearby shows almost no effect. So there's a couple of take homes here. One is that you can kill a lot of birds without being able to detect it. And so data sets like we're collecting at Hotbridge, like are being collected at Veracruz, become more and more valuable. The longer they go, the more data we have against which to compare some of these numbers. Okay, the next one, Diclofenac. So this is an NSAID, non-steroidal anti-inflammatory drug. It's used to have widespread veterinary use in South Asia, where it was killing vultures, old world vultures, by causing visceral gout and kidney failure due to uric acid buildup. If you remember, so here's monocrotophos, LD50, For old world vultures, the LD50 for diclofenac is 1/10 of that. So this is very, very potent for old world vultures. And in fact, the use of this drug was devastating. It was used in livestock for palliative reasons, and the livestock would wander around with diclofenac and die somewhere and then be consumed by vultures. So in a 15-year span, this is a logarithm scale over here on the left. In a 15-year span, populations of old world vultures were reduced by 99.9%. This is virtual extinction, virtual extirpation that occurred as a result of this. Now, we do see signs of recovery following the banning of diclofenac use, but those populations are nowhere near their former size. So let's take another quick interlude and talk about species sensitivity. So why would this be the case? What happened here? Well, in fact, after the experience with old world vultures, people thought, well, let's look at new world vultures, right? Vulture's a vulture. Let's give some diclofenac to new world vultures, like our turkey vulture, and see what happens. And it turns out that at 100 times the LD50 for diclofenac, there was no observable effect on turkey vultures. So it just doesn't do anything to them. And so this is where I think you start to see the effect of phylogeny. Because these new world vultures diverged from the rest of this accipiter clade so long ago, that's obviously plenty of time for differential response to a toxicant to occur. I'll return to that thought in a little bit. But first, we got to talk about mercury. So methylmercury. It enters the environment through manufacturing and fossil fuel burning, where in shallow waters, it is methylated to methylmercury. So it enters as mercury 2, is methylated usually in shallow and or acidic wetlands by sulfur-reducing bacteria, at which point it becomes very bioavailable and indeed bioaccumulative. It's also a neurotoxin, among other things, to birds. And concern over mercury has been increasing for decades. If you look back to the 1950s, this is Google Scholar searches on mercury toxicity birds, you see basically an exponential increase in the number of papers published in scholarship about birds and mercury. And the reason is depicted over here on the side. This is American kestrel data. This is a captive population at Patuxent that was dosed with methylmercury. And you can see that their reproductive output declined pretty severely with increasing dietary mercury. And incidentally, I'll note that several toxicologists from our lab here in Duluth were involved in this seminal study. And I'll come back to that study as well in a little bit. Okay, so some more principles. This is not the same group of exposures, but it's from the same study. The authors developed what's called a Toxicokinetic Model, which describes the time course of uptake and distribution of the chemical into various tissues of the body. We can also define toxicodynamics as the time course of damage resulting from that uptake. And so these authors looked at mercury in the blood of the birds over time following exposure. And you see this big spike in mercury followed by a great decrease. So what's going on here? Well, this is the birds molting. They're pushing all of that mercury into their feathers, basically depurating it. So this would be elimination, right? This is one route of elimination where they're dumping the mercury. These are the model predictions, incidentally. Those spikes there, anybody want to guess what those are? That's reproduction. So each one of those is an egg being laid. And so the model has some mercury being shunted to the egg as well. But overwhelmingly, it's molt that's causing reductions in mercury exposure. So that's good for the bird. The bird can get rid of the mercury. It's also good for us. This is highlighting the work of Ted Kyle, former IBS student here, who looked at mercury in the feathers of many raptors trapped here at Hotbridge Bird Observatory. And one of his overwhelming and most interesting findings was that raptors like Merlin and Shark-Shinpock that eat songbirds are receiving the highest exposures compared to other birds. And so we infer that this is a trophic effect, that the number of trophic levels leading to a songbird is greater, for example, than crickets or small herbivorous mammals. And so that's interesting. But it still leads to the question, so what? How dangerous are these exposures? Well, we use some existing data from the literature to classify the exposures as either negligible, below known effect concentrations, low risk, or moderate risk. And in fact, almost all the birds that we sampled were at low or no risk, but a few were at moderate risk based on the concentrations that we observed. So there are a few individuals out there that are getting enough of a dose of mercury to be of concern. The other cool thing that Ted found is he used stable isotopes of individual birds to assay their individual trophic level at which they were feeding. And so he was able to show that the mercury level in individuals increases with the individual trophic level of that bird. And so we saw trophic effects across species and within species, which is really interesting. So my last act here is the polyfluoroalkyl substances. How many of you have heard of these? What if I said forever chemicals? How many of you have heard of forever chemicals? Even fewer. Okay, that's interesting. It's a room of geeks. Okay, so PFAS are fluorinated carbon chains. They've been used since the 1940s in manufacturing, Teflon, Scotchgard. 3M was a big producer of PFAS. In the 1970s, they started to be used for fire suppression in aqueous film forming foams, or AFFF. And that's really also when they gained widespread exposure in the environment, widespread release in the environment. They are used frequently on air bases, military bases, for suppression of fires in airplanes. And this is actually here in Duluth. This is at Lake Superior Emergency Response Training Center. There's a mock airplane set out in the middle of this circle here that they light on fire and take firefighting foams, perhaps not all containing perfluorinated compounds, and put the fire out. And so this has resulted in widespread releases in the environment. So do we need to care? Some of these chemicals are bioaccumulative, some are toxic, some are both, and most are poorly understood from a toxicological perspective. And note, it's a very large class of compounds, so a lot to parse through here. Wildlife concern began to emerge around the turn of the century when people started assaying wildlife at far-flung places, penguins and seals in Antarctica, albatross at Midway Atoll, polar bears up in the Arctic. All of them were exposed to PFAS. All of them had PFAS in their tissues. And so that was a little bit of a surprise, but then there's still the question of, does it matter? Do we need to be concerned about this? Well, at Hotbridge, we started to get interested and get involved and started a sampling program a few years ago for PFAS. And this slide is just showing you for the moment sample sizes that we've analyzed so far. So far we've analyzed 156 samples for 23 PFAS compounds across these species. And here are our results to date. Note, we don't really see with this particular chemical, this is PFAS, the chemical to which these raptors are exposed at the highest concentrations. We don't really see a relationship with trophic levels right now. So that's a little bit of a head scratcher, but data is still being collected and analyzed, so we'll see. On the other hand, for PFUNA, undecanoic acid, we do see a very strong trophic effect. So different pattern from PFOS, and this leads to the question, oh geez, are these things all going to be independent, or can we use one to predict the other? Can we borrow information to try to understand exposure to these things? Well, this is a principal components analysis that's basically saying, no, we're probably not going to be successful doing that. This is the percent variance explained in the bars of each principal component. And this is the cumulative percent variance explained. And notice that you need about 7 principal components to explain 95% of the data. So there's a lot of independent axes of variation in exposure to these chemicals. More data is coming, and I will note that this is a collaboration we're doing with the Raptor Center down in the Twin Cities as well. We just got a big shipment of samples from them that will be sent out for analysis, and that'll be an interesting comparative data point. So how dangerous is this? We don't know. Stay tuned. We're working on it. That'll be 5 years from now. Okay, so I focused on 5 contaminants, but I've left many stories untold. We could have talked about PCBs, dioxins, lead, neonics, brominated chemicals, cadmium, rodenticides, many others. If you look at those stories, they have a lot of similarities, some differences with the stories I've told. But let's see what sense we can make of what I've got told so far. So the first question we might want to address is, why does this keep happening? Why? And then the second question, of course, is what can Hotbridge do about it? Well, I've given you some suggestions for why this is happening. I think a major one is bad luck, right? The vulture story, who would have known? How would we have known that? That's bad luck. We have also very different regulatory frameworks. So at the bottom of those slides, I was showing you the federal congressional acts that cover these and agencies. And we have lots of different laws with lots of different requirements, lots of different testing requirements that result in a mosaic of knowledge about environmental contaminants. I'm not saying that's necessarily a bad thing. There are reasons for that, but it does result in disparities in knowledge about different contaminants. I've shown you variation in risk among life history and sensitivity and so on. But I'd like to dig a little bit into 2 points now. Let's talk about limited toxicity information. A few years ago, I was curious about this, and I went to EPA's EcoTox database. This is a database that's hosted here in Duluth at our lab. It's curated, hosted, and created. And I said, okay, let's just download all of the data on birds, every single data point, and let's ask what birds are covered. Let's look and see what birds have been studied from a toxicity perspective. And then I classified that by species and mapped that onto this phylogeny here, very greatly simplified phylogeny of the birds. And there's a couple of points that should jump out at you right away. So 6% of toxicity test results in the EcoTox database cover about 95% of bird species. In contrast, about 94% of toxicity test results are conducted on 5% of bird species from just 2 orders, the landfowl and the waterfowl. And to make things even worse, this divergence of the galloensis from neoids happened 72 million years ago. This is 6 million years before the asteroid came and wiped out the rest of the dinosaurs. And so this is an ancient divergence. And it also occurred 14 million years before that New World vulture clade diverged as well. So it's clearly plenty of time for toxicological sensitivity to change. And I think this partly explains why the testing that we're doing down here is not necessarily protective of the birds up here. Okay, what about scientific silos? Also curious about some of these questions. I went to avian journals and toxicology journals, and I asked 2 different questions of each literature stream. I took 4 avian journals and said, how often are they publishing toxicology? And then I went to the tox journals and I asked, how often are they publishing bird studies? And I think hopefully the story jumps out at you right away. Not very many toxicology papers are being published in the ornithological literature compared to what's coming out in the tox literature. There's about a paper a year coming out in 4 main North American avian journals, and about 12 avian papers coming out in the tox papers, in the tox journals. So what else can we say about that? If you look, for example, at the pesticides that are being talked about and published in the avian journals, 8 of 14 studied pesticides in the avian journals were already banned in the US at the time of the study. So that, is not terribly useful from a regulatory tox perspective. It's probably interesting academically. But my point here is to say that these 2 silos perhaps are not talking to each other as much as we ought to be. The toxicologists, we need input from the ecologists, and I think the ecologists really could use input from the toxicologists. And I think these data are an example of that. Okay, so what can Hotbridge Bird Observatory do? By the way, this is the 2021 crew at Hotbridge this year. So if any of you are here, welcome. So Hotbridge really has 2 data streams. We have the migration counts for raptors. Now I'm talking specifically about birds of prey. We have the migration counts, diurnal migration counts that happen up on the overlook. Hopefully you all have been there. If not, I encourage you to go. And we have the trapping and banding effort that occurs back in the woods a bit. And these 2 data streams are differentially useful for ecotoxicological questions. We are contributing, as you've seen, to the Raptor Population Index. I've shown you lots of these kinds of graphs so far. These are the sites that contributed to the RPI in its 2019 analysis. And here is sort of a continental scale picture of what's happening with American kestrel right now based on the RPI. Now, I told you before, don't worry too much about what the y-axis means. And now I'll tell you a little bit about why. The reason is that not every count site counts birds every day, and not every count site has people out there counting for the same number of hours. And so this index has to account for differential amounts of effort that occur at these different sites. Actually, Hotbridge does count every day. So our data are pretty robust in that regard, but other count sites don't. And so you have to do that. But of course, when you're doing that kind of interpolation, you don't know whether you're properly accounting for effort. And so, so that's a potential issue. And it certainly was an issue. Now I'm highlighting Ryan's work, who was looking at whether raptors are delaying their migration. By the looks of the photo, Ryan sure delayed his migration. So the question Ryan was interested in here was whether or not birds were migrating later or earlier relative to the historical record, and further, whether or not changes in effort that have occurred at Hotbridge over the years had any influence on our interpretation of those data. And he found, yes, some birds are migrating later, though perhaps some earlier. This is a complicated story. Ask Ryan about it if you want more information. But not as many as have been reported. And it's because in the 90s, Hotbridge expanded its count effort, which used to extend to the beginning of November. it now extends to the end of November. And that increase resulted in a bolus of birds being counted later in the year, which shifted the mass of migration later. So Ryan showed pretty convincingly that we need to take that into account. You can read more about it in a forthcoming paper. So one of the big questions about Hotbridge, of course, is where are these birds coming from? And of course, one answer is that way, but we'd like to get more precise about what are the geographic locations that are sources for our migrating birds. And historically, traditionally, the way to do that has been to look at band recoveries. This is band recoveries of red-tailed hawks, originally banded at Hotbridge Bird Observatory. And while this is cool and interesting, you can see that almost all these recoveries are south of us. And it's not telling us a lot about where these birds are originating from on their way down. So what other data streams can we use? Of course, I had to embarrass them. I do strongly encourage you to go see this talk tomorrow night in this very same room, 7:30 p.m. It will be awesome, probably better than this talk. But all of these young scientists, these great scientists, are using methods that may help us to tease that apart using indicators of some kind. In this case, Emily's looking at stable isotopes to try to figure out migratory origins. Ali is using population genetics to try and figure out subspecific origins and geographic origins. And of course, Hannah and Ali as well are both using telemetry. All of these sort of newer high-tech methodologies can help us get a better picture of where the birds are coming from. So if we knew where these birds were coming from, and yeah, it's still flashing, and that's awesome. I'm gonna leave that slide up for a long time. If we knew where these birds were coming from, how much more valuable would our environmental contaminants data be, right? We could start at one location to build sort of a continental map of where these birds are being exposed. So keep that thought in your mind. Along with that, another cool study that was recently done at Cockroaches was done by Lisa Boulette, another IDS graduate, who used next generation DNA metabarcoding to try to figure out, using cloacal swabbing, if she could detect prey items in the cloaca of these birds and thereby use that as a method, establish that as a method for studying diet in the birds. And she was successful. She found cool stuff. She largely confirmed that these birds eat what we thought they do, which for a first study is what you want, right? So the methodology does seem to work. There are improvements that can be made to it. So keep that in your head as well. And now let's talk about the Flyway Project. We're basically at the end now. And this, I think, this is a project that we've just gotten underway on with the Biodiversity Research Institute. You remember our Mercury study from here at Hotbridge. We now have tens of thousands of Mercury samples from all of these locations around the United States. And as a group, we will be analyzing those this fall and winter to try to answer questions about the influence of different flyways, the occurrence of mercury in different flyways, different geographic regions. And I think this is a good picture of the vision for where Hotbridge can go with environmental contaminants work, right? If we can expand this and include tracers to understand where the birds are coming from at these various sites, then suddenly we really do have a continental picture of what environmental contamination looks like in these vulnerable birds. So I don't know if that's a year from now, 5 years now, 10, 20 years from now, but I think it's coming, and I think it will greatly enhance the value of research, not just at Hotbridge, but at other banding stations as well. So Hotbridge is no longer just an autumn station. We do a lot in the autumn, but there's lots going on. We have work on population cycles. Dave, are you out there? There he is. Yep. So that's Dave Shemp's work, and Dick Green is working on this with Northern, sorry, not Northern Harriers, Northern Goshawk. And we have lots of other stuff going on. I want to highlight this one. I think is really cool. We've just gotten into a study of mercury and oxidative stress, where we're actually looking at the bioresponse of some of these birds and being able to relate it to the mercury burden that they will be carrying as they fly through. But we now have stuff going on in winter. This is Hannah and others doing winter owl work. We've got spring and summer, and I think Hotbridge is really expanding to an annual organization at this point. I have to come back to my theatrical beginnings, right? Hotbridge really only exists because people are passionate about it. It has happened because people, some in this room, had the foresight to set up Hotbridge and to realize that this was a phenomenon that needed documentation. And I wanted to highlight several people behind the scenes: David Alexander, Abby Valine, Miranda Durbin, who really, the environmental contaminants work here would be nowhere without them. And I will also highlight Frank Nicoletti here. He is our Zeus, perched atop Olympus there, day and night, it seems. And yes, none of us doubt that he could strike us down with a thunderbolt at any moment. We exist because these people, many mentioned here, Jan and John, I see in the audience today, had the foresight to make this happen. We also have gotten a lot done through our partnerships. So we are people, partners, and birds. So if you're any of these three things, you're welcome at Hotbridge. And if you have a great idea, we'll help you make it happen. So thanks. Lots of people have funded us, especially Hotbridge members. And many of these grants are to our great graduate students. So I'll acknowledge them here too. So thank you for listening. So do we still do undergrads first? Is that okay? Questions from undergraduates?
So I noticed you're working with different labs for the US, but are you working with any in Canada or farther north? Then you mentioned one or like where the birds are coming from.
No, we're not. That's a good point. We don't currently have any international collaborations. We would be, we'd love to do that. Interestingly, all of our contaminants data are being sent to Canada for analysis in Vancouver, but that's as close as we get to a Canadian cooperation right now. But we'd love to do it. I think it'd be a great idea.
Why do you think there's a disparity with like 94% of the toxicological studies on landfall waterfall?
That's a great question. So there's some really good explanations for it. One is a lot of those birds have been domesticated, and they're really easy to deal with in pens. They'll lay eggs when you want them to lay eggs. You change their light regimes. They'll go into breeding condition. That's really hard to do with an albatross. And so there's a lot of really good reasons for it. I didn't mean that to sound pejorative, but it is there is a bias that results from that. Any more undergraduates?
What would be if you would get involved with this research?
Oh, yes. Talk to one of them. Absolutely. Come up to the Ridge, tell somebody you're interested, send me an email. If you know any of this motley crew over here, send them an email and we'll plug you right in.
I will say, for any interested third undergrads, there is going to be a new student-led organization for a UMD bird club. So stay tuned. If you're interested, come talk to us.
Awesome. Any more undergrads? Going once? Going twice? Grad students? Okay, let's open it up to anybody if there are any last questions.
Well, just following up on that question about Canada, are there good migratory observation stations up in the area where you would most like to get the data?
Way up north, no, but I mean, like everything else, all the other population density in Canada, it's kind of clustered along the border, but fewer up north. These guys might know more about far north stations. Are you aware of any way up there?
Alaska,
Alaska, yeah.
but that is.
That's kind of coastal. Yeah. Way in the back.
So I don't know if I heard it well, you might have said this on the phylogeny case, but is there some conservative form of phylogeny to different responses to these different toxins? So in other words, is there phylogenetic signal to that?
That's a good question. I think there is. The strongest phylogenetic signal that I could see was in that Diclofenac story, where you see, that really early branch. In that case, conferring resistance, not sensitivity. The question then would be: it looks like the old world vultures are nested in with the accipiters and bootios, and so then the question is: are the accipiters and bootios equally sensitive? What does sensitivity look like there? And I don't know the answer to that question. That would more closely address your interest. Yeah.
And somewhat follow up to that, are there model systems where there are breeding programs of raptors for experimental studies?
Yeah. So the Kestrel study is probably the best example of that. That's been going on for quite some time. And in fact, so I mentioned PFAS. There are PFAS studies with Kestrels getting underway now. So. Yeah.
How did you go from math to bird?
Still asking myself that question. Through a series of bad breaks and misunderstandings, I guess. No, I always loved birds, except when my parents took me birding, which I hated. But I ended up in Africa, as Jerry mentioned, for Peace Corps, and did a lot of birding there, a lot of cool birds, and then from there, traveled my way around the world to get back and birded all the way, and I was pretty much hooked at that point. But I knew I wanted to do math birds, not just plain birds. I wanted to do math. Yes, exactly. Any other questions? Yeah.
What's your favorite bird?
Oh, that changes from day to day. Right now what pops into my head is peregrine falcon. But Merlin is an amazing bird too. I guess the falcons I'm disposed to like the falcons. Yeah. Anything else? All right, well, in that case, let's all thank me for a great talk.